Good afternoon, everyone. So my name is Tristan and I'm Dario's PhD student. So for today's presentation, I'll be talking about perhaps a different topic to what most of you may be used to. So my research is really on ultrasound rather than EMG. And I'll be talking about the development of ultrasound as a human-machine interface with the central nervous system and the musculoskeletal system. So for today's presentation, I'll first go through a bit of background behind this work and then describe some preliminary results that have been obtained for two studies. First, the development of ultrasound as an interface for the central nervous system, and secondly, the development of ultrasound as an interface for the musculoskeletal system. And finally, I will conclude. So let's first go into the background behind this work. The human-machine interface market has been growing, and it's projected to grow from 6.7 billion US dollars in 2024 to 7.4 billion US dollars in 2034. And this represents a projected compound annual growth rate of approximately 10.6%. And ultrasound has the potential to play an important role in some of these devices because of its advantages, such as having a relatively high spatial temporal resolution, a high penetration depth, a large volumetric coverage, and minimal to no invasiveness. At this point of time, however, ultrasound remains more of an imaging modality rather than an interfacing modality, and this is perhaps because of the history that biomedical ultrasound has played in medical imaging. And so in this work, what I endeavor to do is to develop ultrasound as more of an interfacing modality rather than an imaging modality, and I'll come back to explain a bit more of what this means. And the applications I'll be looking at will be the central nervous system and the musculoskeletal system. So let's first now go into the development of ultrasound as an interface for the central nervous system. In this case, ultrasound is a brain-machine interface that leverages the principle of neurovascular coupling. What this means is that because of the tight connections between neural cells and blood vessels in the brain, the hypothesis is that when neuron cells fire, there's a corresponding increase in blood flow to the associated region within the brain. And thus, by measuring the blood flow, which ultrasound can do, we can indirectly make observations of neural activity. And this has advantages over the different types of brain-machine interfacing technologies that there are out there. So, for example, compared to EEG, ultrasound has a higher spatial resolution. And compared to functional magnetic resonance imaging, it has a higher temporal resolution and a higher accessibility because of the portability and the more affordable nature of ultrasound systems. Compared to functional near-infrared spectroscopy or FNIRS, which is also another portable brain-machine interfacing technology, ultrasound systems can provide a higher penetration depth and allow the interrogation of deeper brain regions. And finally, compared to electrocorticography or ECOG and intracortical electrodes, ultrasound has a low invasiveness and can also provide a higher volumetric coverage. So just to go over a bit of the literature behind the use of ultrasound as an interface for the central nervous system, the first report really started in 2011, and this preceded a set of papers that were published using animal models and observing the ultrasound responses to various kinds of somatosensory stimuli, such as whisker, electrical, auditory, visual, and pain stimuli. And so in all these papers, what was observed was that neural activation in response to these different stimuli could be detected using ultrasound readings. And more recently, this has been demonstrated in a human while he was playing a video game and a guitar. So I've pulled some images from this publication and they are shown here. And if you see in figure A, this shows the subject with an ultrasound probe fixed to his head while he was playing a video game. And figure B shows the experimental protocol that was adopted, where there were alternating periods of rest and while he was playing a connect the dots video game, or what we would term as stimulation. And so what the authors did was to record ultrasound images of blood flow in the brain during these two periods of rest and stimulation, and then construct a statistical parametric map to observe any significant differences in blood flow patterns between the rest and stimulation periods. And what they observed was that there was a difference between rest and stimulation, and thus that these two periods could be distinguished. So what was remarkable was that this individual, as you can see in figure A, was actually someone who suffered from traumatic brain injury. And he was actually had a skull replacement and was on the road to recovery. And so it showed that ultrasound technologies could be used as a rehabilitative or assistive technology in brain-machine interfacing. However, at present, there still exists a gap, and that is that in all the studies that were shown in the previous slide, there was the need to reconstruct and analyze a functional ultrasound image. Now, this is time-consuming and computationally intensive, especially if high resolutions are used. This means that it's impractical for use in closed-loop brain-machine interfaces, which require the fast processing of information for the quick actuation of end effectors. And so the proposed solution that I had was to use neural networks on the raw ultrasound radio frequency data to provide a more direct interface. To implement this, what was conducted was a study where there was one craniotomized and anesthetized rat which was exposed to two kinds of somatosensory stimuli, auditory and whisker stimulation. And all this while ultrasound recordings were being made. And so for the different kinds of stimulation, there was 3 alternating cycles of rest and stimulation. And in cases where microbubble contrast agents were used and not used. And this was also applied for auditory stimulation. And so once all the ultrasound data was obtained, this was put through a convolutional neural network, which consisted of 3 convolutional blocks that had convolutional and max pooling layers, and into a flattened layer, and finally a block of fully connected layers to output a value of 0, which indicated the absence of stimulation, or 1 to indicate the presence of stimulation. So the results from this were very promising for each stimulation type across the training and validation and test sets. And what can be seen is that the classification accuracies exceeded 90%, which was equivalent or even better than some of those obtained when an image was reconstructed. Now, what's more impressive is that the time taken to classify a single image in each of these stimulation types was 59 milliseconds. What this means is that from a period of more than 100 milliseconds, which is the time taken to reconstruct and classify an image that's obtained from functional ultrasound, we can now obtain a classification of in 59 milliseconds using the raw radio frequency data. Now, this is promising because it can pave the way for quicker actuation of end effectors in closed-loop brain-machine interfaces. But future work for this to be realized would involve obtaining data from a larger sample size and perhaps including more kinds of somatosensory stimulation in order to perform multi-class classification. We can also experiment with different kinds of model architectures and hyperparameters, and finally investigate the explainability of some of these models. Now, moving on to the development of ultrasound as an interface for the musculoskeletal system. The background behind this work is that ultrasound imaging is very commonly used to examine the internal anatomy and function of muscle tissue. So this is an ultrasound image that was taken of the tibialis anterior muscle, and here we can see the annotated in the image the superficial and deeper connective tissue layers, as well as the single muscle fascicle, which is represented by the red line. And so 2 parameters that are important to measure in this image would be the fascicle length, because this influences the amount that muscle fascicle can shorten and lengthen during a contraction, as well as the pennation angle, which affects the dynamics of contraction through influencing the length, tension, and force velocity relationships at the tendon. So the length of the red line represents the fascicle length, while the pennation angle is represented by the angle that the red line makes with the green line. So There again exists a gap in that conventional ultrasound imaging systems are bulky and they have wired connections because they require a connection to a power socket. What this means is that it impairs the natural execution of movements such as walking and running, and also that these ultrasound systems are very often limited to lab settings. So if you can see in this image here, this was taken from a publication in the field of the setup they were using to measure the fascicle length and pennation angle of the gastrocnemius muscle in the calf. So as you can see, they require a very bulky setup with foam to fix the probe to the muscle as well as a cable. And this perhaps is useful for small contractions such as calf raises, but perhaps not very practical for movements such as walking or running in the real world. And so the proposed solution to address this would be to measure these quantities of interest, the fascicle length and pennation angle, using single element transducers, which work on the principle of A mode or amplitude mode ultrasound. The advantages of these would be that with fewer transducers that can be used, it will represent a smaller form factor, which would make it more wearable, and also require fewer computational resources to analyze the data. So to implement this, what we did was to use a conventional B-mode imaging probe and single element transducers in a 3D printed housing setup on the tibialis anterior muscle, which is a pennate muscle, to measure both B-mode and A-mode data at the same time to see if the A-mode data could observe what the B-mode data could. And so 5 single element transducers were placed on either side of the imaging probe while participants performed the task. And that task was alternating dorsiflexion and plantar flexion to vary both the fascicle length and the pennation angle of the muscle. So this was done for a duration of 30 seconds across a range of frequencies from 0.5 to 2 Hertz, which represented the range of frequencies in walking and running. The number of subjects analyzed for this was 2. So once the B-mode data was obtained, the sequence of images was put through a tracking algorithm, which tracked the fascicle length and the pennation angle across image frames to produce the graphs that you can see below. And so I have a video to show the tracking procedure, and I'll just play it. So as you can see, the ultrasound image can observe the muscle contracting. And as it's contracting, the fascicle length, which is the length of the red line, and the angle that it makes with the green line are being recorded. So once this data was obtained, the fascicle length and the pennation angle data were used as the ground truth, while the radio frequency data obtained from the single element transducers was used as an input to train another convolutional neural network consisting of, again, 3 convolutional blocks, which had convolutional batch normalization and max pooling layers into a flattened layer, and finally a fully connected block to output a value for fascicle length or pennation angle. So this was a regression type model. And the results of this for the 2 subjects, we use subject-specific models here because of the insufficient number of subjects for a generalized model, were quite promising as well. And for both subjects, what we could see is that the root mean squared error across the training, validation, and test sets for the fascicle length was in the range of 5 millimeters, while that for the pennation angle was in the range of 2 degrees. And this represented errors which were less than 13%. And so what this means is that A-mode ultrasound, rather than B-mode ultrasound, has the potential to measure both the length of these fascicles and the pennation angles that they make with the connective tissue. And this is advantageous because the single element transducers occupy a smaller and more wearable form factor and cannot be computationally lighter. So to improve this, future work can again involve obtaining data from more subjects and recording the A-mode and B-mode data from the same place, because a key limitation of this study is that both A-mode and B-mode data cannot be recorded from the same place at the same time. And in this study, we chose to record the two types of data at slightly different places, although adjacent places at the same time. But an expansion of this work can include recording the data from the same plane, albeit at different times. Again, we can also experiment with different kinds of model architectures and hyperparameters and investigate the explainability of these models. So to conclude, what these two preliminary studies showed was that deep learning models can enable anatomical and physiological quantities of interest, such as brain state, fascicle length, and pennation angle, to be determined directly from ultrasound radiofrequency data. So in the past, when ultrasound was more of an imaging modality, what would be done is to obtain radiofrequency data, to reconstruct an image, and finally obtain a target output. But now what we have shown is that the target output can be directly obtained from radiofrequency data, and this is what we deem as the development of ultrasound as an interfacing rather than an imaging modality. So this is beneficial because radiofrequency data contains all of the information that is available from an ultrasound acquisition. In the process of using this radiofrequency data to reconstruct an image, the pre-processing steps along the way result in the loss of some information or features, and this perhaps may be detrimental to the extraction of a target output. So using all of the information and learning the exact features to extract is perhaps beneficial for the prediction of a target output. Secondly, the algorithms developed to analyze this radiofrequency data rather than an image may be computationally lighter because they pick out and learn only the exact features from the radiofrequency data that is useful for the prediction of a specific target output. And finally, this means that smaller and more wearable forms of ultrasound instrumentation may be available for bodily analysis. So with that, come to the end, and I'd like to thank you for your attention.